Downstream processing refers to the recovery and the purification of biosynthetic products, particularly pharmaceuticals, from natural sources such as animal or plant tissue or even the microorganisms. This also includes recyclable of salvageable components. Like salvageable components means that those components which are considered as waste, but they do have some applications. So recovering of those are known as salvageable components. And also downstream processing is also used in the proper treatment and disposal of the waste. So downstream fermentation is the last process of your fermentation technology. That is, the product, whatever you have extracted, it has to undergo purification. So this, the end portion, what you're getting, that is known as the downstream processing, and this has various steps wherein where we use analytical techniques. Now, if we consider microorganisms, the microorganisms metabolites means which are the products which are useful to us. Okay, these metabolites can be produced by the microorganism intracellularly or extracellularly. So here, or in some organism, what happens, it is both intra and extracellular. Now, how are we going to extract these products from the microorganisms? How we are going to extract it, if it is intra or extra or both intra and extracellular? That is what we will be looking into it. So there are different stages of downstream processing, such as you can see here, separation of particles, disintegration of the cells, extraction, concentration, purification, and drying. We shall study each of these topics individually. So the first one, what we told, is the separation of the particles. This is also known as solid liquid separation. So this is the primary recovery operation. So usually here, what we are going to do is, the first step in the product recovery is the separation of the whole cells. The whole cells means the microorganisms or even the plant or even the animal cells. So to avoid confusion, I will use only microorganisms. Okay. So the separation of the whole cells and the insoluble ingredients from the culture broth is known as the separation of particles. So in the fermentation, as we have already told, we will be adding what all the nutrients what is required for the organisms, the physical, chemical conditions. So here what happens in the fermentation? Now your product, your substrate, your microorganisms are all mixed up. I don't want everything. I just want my product. And that phenomena is the separation of the particles. So these particles, okay, some of the particles are insoluble ingredients. For matter, as I told you earlier also, some of the metabolites are produced within the cells of the organisms, or so what you have to do, it must be released from the cells before subjecting to the solid liquid separation. So let me write it and explain to you all. See, some, if this is a microorganisms and if this is your fermenter, the metabolites can be secreted externally. But what if it is produced internally? Then internal separation requires a more tedious process, okay? Because I have to break open the cells of these microorganisms and then extract them and purify them. So there are different steps to be done to extract the metabolites. These can be like filtration, centrifugation, flocculation, and flotation. So the first procedure what we are going to look into is the flocculation. Here what picture you're seeing here, this is known as the flocculation. This is the flocculation of the yeast sample. So in flocculation, the cells or the cell debris form a large aggregates, okay, to settle down for easy removal. So the process of flocculation depends on the nature of the cell and the ionic constituents of the media. Addition of flocculation agents like such as the inorganic salt or your organic polyelectrolyte, mineral hydrochloride, hydrocolloid is often necessary to achieve this flocculations. So what is happening here? All your microorganisms are forming a flock or they are sticking to each other so that they can be removed easily by the other principles like filtration and centrifugation. Now, the microorganisms has to get flocculated, okay, like this. They has to adhere to each other. So what the scientists do is they add certain compounds. That compounds can be inorganic or the organic salts which have already mentioned, such as here have to like polyelectrolytes, okay. So these flock may float to the top of the liquid to form a cream. Otherwise, they may settle at the bottom of your fermenters, which is called as sedimentation, and they can be filtered off very easily. The next step what we are going to speak is the flotation. Now you can see here at the picture. Now, what happens in flotation is when a gas is introduced into the liquid broth, it forms bubbles. The cells and the solid particles get absorbed on the gas bubbles. So these bubbles rise to form a foam layer which can be collected and removed. The presence of certain substances referred to as collector substances facilitates stable foam formation such as the you can add long chain fatty acids and amines. So the main important here what you are going to do is you have look at this structure, the diagram. Here you have the inlet air, okay, where we can pass the gas. So as I have mentioned, the gas is being passed, and what happens to the gas? The gases form tiny gas bubbles onto which your cells means the microbial cells gets attached to it, okay, and then you can collect them by using the collectors as I have mentioned, like long chain fatty acids. So what they are going to form once these microorganisms bind to your gaseous substances, they are going to form a froth or the foam, which can be removed by the simple method. Next, coming to filtration. As you all know, filtration is the most commonly used technique for separating the biomass and culture filtrate. And the efficiency of filtration depends on many factors. It depends on the size of the organism, the presence of other organisms, the viscosity of the media, what temperature you have dealt with your fermentation. And there are several filters such as the depth field, absolute filters, and rotary drum vacuum filters, membrane filters. Of course, we'll not go into all the filters, but we shall look into the membrane filter. So, filtration, it is used to separate your biomass and the culture filtrate. So what did I tell? As I told earlier, it depends upon the size of the organisms or if any other microorganisms are also present. So we have what is, I told you, the membrane filters. So these um, membrane filters are very useful technique in this. Particularly the separation of the filamentous fungi and filamentous bacteria, as I mentioned, like streptomyces and yeast, they all usually form clots. In membrane filters, what we are going to do is, in this type of filtration, membranes with specific pore size can be used. So now it is, what did we tell? It is a membrane filters. So suppose consider this is a filter. So obviously you know that the filters will be having pores. Okay. And this pore size will vary. It can be of different sizes. However, clogging of filters is the major limitation. So when you have these filters with the pores, when you are adding your material into it, what happens? You want to collect all your unwanted products into this, and so that your purified products comes out, the filtrate comes out. But now when what happens is when you're adding the product, you have the pores and the pores can get clogged. That is, it can get blocked. That is one of the major limitation of filtration. Otherwise, it is one of the good technique. You have what is known as cross filtration, wherein the culture broth is pumped in a crosswise fractions. Okay, like it will be in the crosswise fraction you can collect. So this, when you're collecting in the crosswise fraction, what happens is this reduces the clogging process, and hence it is better than the other filtration like static filtration. Static means you know that it is in one position. So we always, it's better to go for cross filtration so that there is no clogging of our filters. What I mean by clogging means that the pores of your filters are not blocked. Next, we come to the centrifugation. So this is also the, we are still dealing with the first part of the downstream processing. Now in centrifugation, what happens? First, the technique of centrifugation is based on the principle of density difference. Okay, this point you have to remember. Whenever you're doing centrifugation, how it is getting separated, it is because of the density. There is the difference between the density of the particles present in your samples. Thus, centrifugation is mostly used for separation of solid particles from the liquid phase. Unlike centrifugation that is conventionally carried out in the laboratory scale, there are certain limitations for large scale industrial centrifugation. Now, in the laboratory, we use small centrifugation. That is, you know that it is obviously not useful for the large industrial scale. But nowadays, in what has happened is there is continuous flow industrial centrifugation have been developed, and there is continuous feeding of slurry and collection of the clarified fluid, while the solid deposited can be removed intimately. So now what does this mean? It is an automatic process is occurring, okay, wherein there is a continuous flow of the industrial centrifuge. So whatever the products you're getting, directly it goes into the centrifuge. The centrifugation occurs, and whatever the material you want, the important material, that can be easily removed. And also there are different type of centrifugation, which we are not going to go into detail of it. So one of the drawback of this centrifugation, of course, it is expensive, but it is one of the best methods. Okay, as I told you, since now industrial centrifugation is being developed, the entire process is becoming easy because there is, as I told you before, there is a continuous feeding of your slurry and collection of the clarified fluid. You keep adding your materials which you want to sediment. Based on the density, they get sedimented, and what happens, you can collect your fluid material, and the solid material get deposited here. You can see that. See here, you can see it here very easily. The solid materials are getting deposited, and the supernatant is the liquid. So this is an automatic process which can happen. Then the use of centrifugal force for separation of mixture. What does this mean? Means now how the centrifugation operates is, it is because of the centrifugal force. Because of the centrifugal force and because of the density of your material, that is the separation of the mixture. So more denser your compound, or when your compound is heavier, they migrate away from the axis. Look at this. This is the axis. Now, when a compound is very heavy, what happens? They move away from the axis. And if it is a lighter substances, the components migrate towards the axis. So this is the technique how the centrifuge works. So the most important point is the centrifugal force and the density and the what is the RPM you are using. So you will get a supernatant and you get the pellet. And depending on whether you want to collect your pellets or you want to collect your supernatant, you can separate them. Next, we have what is known as the disintegration of the cells. So, in disintegration of the cells, what we are doing is that we are disrupting the microbial cell to break the cells in order to release its contents of the cells into the media. So, as I told you previously, if this is the microorganism and all your intracellular metabolites are present in this, these cells have to be broken down so that you release these materials which are useful for us. So disruption of the microbial cell is difficult to do. Why? Means because of the small size of the cells, and strong cell walls will be present in some of the plant cells and microbial cells, and we have to use a high osmotic pressure. So we can look into the different type of equipments which are used for disrupting the cells. First one is the ultrasonication. Ultrasonication is a method, or it is a process wherein you have an equipment wherein you add your material of interest into the ultrasonication. Okay, so it is, for example, if it is like this, it is an ultrasonication, and here what we use is we use sound waves. Okay, it is an ultrasonication, so we are going to use sound waves to break the cell walls of the plant or the animal cells. Otherwise, what you can do is you can go for the osmotic shock. So the cells are suspended in a viscous solution like 20% sucrose, or it could be glucose. Then the cell suspension is then transferred to the cold water, that is 4 degrees centigrade, which results in the cell lysis. So this is known as the osmotic shock. Okay, why osmotic shock? Because there is sudden shift from the normal temperature to 4 degrees centigrade. Okay, because of this there is the destruction of the cells. So how we are going to do this? We will suspend our cell particles in 20% of the buffered sucrose, or you can use even the glucose, and then you transfer the cells into the cold water. What is the temperature? The temperature is 4 degree centigrade. And because of the sudden change, what happens? The cells are facing a sudden shock, and because of this osmotic shock, you can disrupt the cell, or you can break the cell and release the component. Another method is the heat shock, which is known as the thermolysis, where we subject the cells to the heat. Okay, the cells are exposed to heat, which results in the disintegration of the cells. It is an economical method. When you're using heat shock, it is very economical method, but a problem is that if your product cannot withstand heat, you cannot use a heat shock, or thermolysis cannot be used. Lysis means you know that it is to break. Next, we can use even high pressure homogenization. This is the high pressure homogenization. First, we look into the theory part of it. What is meant by high pressure homogenization? It involves forcing of your cell suspension at a high pressure through the narrow orifice to come out to the atmospheric pressure. The sudden release of pressure causes the cell breakage. So now what has happened? There is a small orifice which is being present, and into which your cells are forced into, and there is a very high pressure in which, because of this, there is an breakage of the cells. So you can see here, this is the pressure. Okay, and these are your cells, and this is your that small orifice through which they come out. When they come out, what is happening? You're getting the homogenized particles. Okay, see, the particles are all so small. That is, the cell destruction is happened because of your high pressure, what you are implying. The next one, what we have is the in pigment. Here, the cell suspension at high velocity and pressure are forced to hit a stationary phase. So what happens? You have a stationary phase, okay? And you have very high velocity and pressure. They are forced to hit the stationary phase. Who? Your cells, okay? So the cells are disrupted by the forces created by the end point of the contact because they hit so forcefully because of the velocity and because of the pressure that the cells are broken. And once they are broken, whatever the product you wanted from the intracellular of the cell, they can be removed. Apart from this, we can use even the chemical methods. In chemical methods, we can use detergents, okay, to disrupt the structure of the cell membrane by solubilizing their phospholipid. So, what you all know that the microorganism cell membrane consists of what? Phospholipids. So, obviously, we'll be using detergents to look into that. We will break the cell membranes. Okay. So, this is again like lipid, protein, and protein interactions. With that, we can bring about the breakage of the cells. So these chemicals are mainly used to rupture particularly the mammalian cells. For disrupting the bacterial cells, detergents have to be used in conjugation with the lysozyme. And with the fungal cells, for example, if you're using yeast and mold, the cell walls have to be similarly weakened before detergents can act. So what happens is you can use the detergent. Of course, you're going to use the detergent, and sometimes you have to use the lysozyme to break the components. So, what are the detergents we are going to use is the ionic and non-ionic detergents are used. So, there is disadvantages also. It affects the purification step. Mainly, the salt starts precipitating. For example, Twin and Triton X 100. Apart from this, we can use even the organic solvent to bring about the cell destruction. So the organic solvents mainly act again on the cell membrane by solubilizing its phospholipids. So if I'm having if this is the microorganism, you know that there is a phospholipids present beneath the, in the cell wall. So what we do is, in order to break these, we can use even the organic solvents. Like what are the most common organic solvents we use is the like toluene, Toluene. Toluene are more commonly used to disrupt the fungal cell walls. You know that the fungal cell walls contains the chitin. Okay, it is difficult to break them, so you can use this. And even the other solvents are used like that of alcohol, ether, DMSO, benzene, chloroform, etc. also can be used. So again, we can use what both acid and alkalis also. Apart from detergents and organic acids, we can use even the acids and alkalis because it is the easiest and least expensive method available in the general laboratory. And the method is also very fast. It is reliable and relatively clean way to isolate particularly the DNA from the cells. And it can be used for both laboratory and the industrial scale. For example, like alkalis, we can use NaOH. And lastly, another important technique is the enzymatic lysis. In enzymatic lysis, what we do is the cell walls are broken down by using the enzymes. So previously we told that we are going to use alkalis or acids or organic solvents or detergents. Apart from these, even you can use enzymes. So what is the enzymes we use commonly is the lysozyme. So this enzyme hydrolyzes the beta 1,4 glycosidic linkage. Okay, beta 1,4 and for glycosidic linkage of the peptidoglycan layer of the bacterial cell. And if it is a fungal cell, they can be lysed using enzymes like chitinase, cellulase, and mannase. So enzymatic lysis, you can use it for both the bacterial and the fungal. But of course, there are different enzymes what you use. As I said here, particularly the lysozyme, you use it for the hydrolysis of beta 1,4 glycosidic bond, which is present in the cell wall of the bacteria, that is the peptidoglycan layer. And gram positive bacteria are more susceptible. Okay, you can easily cleave the gram positive bacteria. And the other enzymes what you can use is the glucanase, mannase, and protease. So this is the enzymatic method. One more method we have that is known as the grinding with beads. So what is this grinding with beads is? I'll show you the picture in the next slide. It consists of a cylindrical body with an inlet and it will be having an outlet. So there is an inlet and there is an outlet and there is a motor driven shaft. So that this motor driving shaft is fitted with an agitator. Okay, that agitator can rotate. So the filter is fitted with there are glass beads which are present. Okay, so if it is like this, you have the motor. The motor helps in the rotation of the agitator, which is attached to this motor, and the cylinder is fitted with the glass beads. And when there are cells, microbial cells which are present, so what happens, the cells break as they are forced against the walls of the vessels of the bead. So the beads crush the cells. So what happens, the cells are disrupted. You can see here, look at this. Okay, so these are the rolling beads. So what happens? There is an agitator which is present, which keeps rotating. So your beads and the cells are rotating. The cell suspension containing glass beads is subjected to a very high speed in the vessel. So the cell break as they are forced against the walls of the vessel by the beads. So these are your cells being disrupted, this region what you are seeing. And these are the beads which are rotating. You can see the symbol.